Welcome to Chapter 6, Nuclear and Cytoplasmic Staining. Now, as I mentioned in the previous chapter, we're probably going to be doing a lot of drawing from this point forward. Mostly because, as we'll find out, in histology, when you do staining, you're kind of adding layers of color to things and sort of washing out the things you don't want. So I find that kind of drawing our stains is a great way to illustrate how they work. And so, as I'm using my drawing software here, I would encourage you, if you are a interactive kind of person, that you would draw along. And how you can do that, you can use the same software I'm using. It is called Krita. That's Krita. And it's a free software, and I know it works on Windows PCs. I'm not sure how it does for Mac. But really, any kind of image editing software that allows you to have layers. You can see over on the right side here them changing between. Adding layers is a really good way that allows us to kind of separate things, not color over things we don't want to affect. So you don't need Photoshop or anything high-end. Really, there's a lot of free options out there that you can use. So yeah, let's get started. So for nuclear and cytoplasmic staining, there's quite a lot of theory to go over before we really get into coloring things and kind of get into the fun stuff. But it's absolutely crucial that we understand how dyes work and kind of the structures of cells, because tissue is just a collection of cells with a similar purpose. So we're going to be ultimately looking at a lot of different cells from a lot of different tissue types, but it's just going to be we're going to be seeing a lot of cells individually as we're looking at those tissues. So let's just start. We're just going to draw one circle. You don't have to worry about making a perfect circle because, gosh, trying to find a cell that is a perfect circle is kind of hard. Because on the outside here, this is all phospholipids. So there's actually two layers of phospholipids. You can just kind of think of those as lipids or fats, and that's kind of what keeps things inside the cell or things outside the cell that we don't want to be on the inside. Next, we're going to be looking at probably one of the larger structures that you're going to see in a cell, and that's the nucleus. And it tends to be where all of our DNA is stored. When you're looking at a nucleus, it might look a lot like this one, where it's very solid and there's only one of them kind of occupying one spot, and that tends to be in cells that are in between phases of cell division, so that this nucleus and this entire cell is in a phase of interphase, meaning that it's not trying to divide right now. It's just kind of doing its other duties as a cell wherever it is. So it's probably making a lot of proteins and doing other work inside the body. So ours is very, very stable right now. And if we wanted to look at the cell cycle, then we probably start looking at different shapes of nuclei as they go through that. But for right now, we're just going to work with an interphase cell. Now, inside that nucleus is a concentrated spot, which sometimes, if your H&E staining is just right, will actually be red. And we'll get to why that is here in a few moments. But that is our nucleolus. And before we start adding color to anything, this is going to be a lot more along the lines of what you might see on an electron micrograph compared to light microscopy, where we rely mostly on the dyes to show us where everything is. The rest of our nucleus is mostly going to consist of some different amounts of chromatin, which is mostly just our DNA. It tends to be at varying stages of being wrapped or kind of coiled in on itself. So if we look over here, this one is very tightly wound, and that is our hetero chromatin. Let's put an H there for that. So our heterochromatin is stuff that is not actively being used or duplicated. And so it tends to be a little bit more dense, and it also tends to stain. So when we get into our nuclear staining, that's the part that you're mostly going to see showing up. And then this loose stuff over here is actively being replicated, as we know when we're creating proteins, that the DNA has to be unwound somewhat. And so that is our euchromatin, which is genetically active and does not stain all that well. Once we get outside of the nucleus, then we start to see some very dense looking lines that tend to kind of move out from the sides here. And those are endoplasmic reticulum. Now, the endoplasmic reticulum comes in kind of two flavors. Some are very smooth, so this would be our smooth ER, and some are pretty rough. That's our rough ER. And the rough ER tends to have these little dots on it. And what those little dots are ribosomes. And so between the ribosomes and the rough ER, protein is produced by the ribosomes by taking the mRNA from the nucleus and turning it into a protein. And then the ER that it's attached to, making it the rough ER, will take that protein and package it and move it either outside the cell or to different parts of the cell that need it. So that's much more about moving those materials. Now, that's opposed to the smooth endoplasmic reticulum. Smooth endoplasmic reticulum is used mostly in lipid synthesis. So it's kind of going to create lipids and then take a piece of that ER and just kind of move it somewhere else to move those lipids, typically to repair things like the plasma membrane or other parts of the cell. Also situated fairly close to the nucleus are your Golgi apparatus. Now, these are probably going to look kind of like your ER, except they're going to be kind of in plates or stacks sometimes. And the role that they play is in transporting things. So we said that our rough ER is making things kind of move outside of the cell sometimes. So it will send things, typically proteins, things like that, over to the Golgi apparatus. And the Golgi apparatus will take those things, those proteins, and make them into vesicles, which are just these little balls that are used for transporting. And those will move those out to the plasma membrane where they can be secreted. And they typically do that by kind of adhering a little bit to the membrane and kind of just folding out and pushing all the contents out of the cell. So that's your Golgi apparatus. You don't typically see it on an H&E stain, but it's there. Also on an electron micrograph, you might see some little oval-shaped figures, and those are mitochondria. Your mitochondria are typically known as the powerhouses of the cell, where they create ATP, which is then used for energy. You won't see them on an H&E. These are things you're going to mostly see on an electron micrograph. Another circular or oval-shaped object you might see on an electron micrograph is a lysosome. And lysosomes tend to be packaged with things that are used to digest other things. So you might use it for defense if you're trying to phagocytize an invader, or if you're just trying to digest food particles, depending on what the cell's role is there. That's typically where you're going to find all those little enzymes and different things like peroxidase. Something else that you might come across, there are a couple of long-looking things. There are a couple tubes, and those are centrioles. And they're typically used in creating a spindle during cell division, but outside of cell division, they're kind of just sitting here, tend to be perpendicular to each other's long axis. They kind of make an X. And you can't really see them outside of an electron micrograph. That's about it for the ultrastructures found inside the cell. So we're going to go ahead and clean this up. And if you're following along, I'm just going to go over here and remove that layer that I was drawing on. See how it just kind of makes it nice and blank for me. And I'm going to create a new layer to draw our new cell on. So we're just going to draw a new cell. We're going to be talking about the cytoplasm and the nucleus in terms of staining and different types of charges and all of that. Before we get too deeply into that, we should probably talk about bonds. We're talking mostly about chemical bonds here. So one of those that we might come in contact with is ionic. An ionic bond tends to be a positive and a negative. So you have one substance or a molecule that's positive, and it's attracted to a negative one. These tend to be very pretty strong forces, and they're mostly just held together because the charges are opposites. Then we have hydrogen bonding. And that tends to be between, have a covalently bonded hydrogen. So this isn't a water molecule, so we have our oxygen up here. And those are attracted to something else that has a high electromagnetic charge. And this can be found, again, in water. So we have another oxygen here. It's more hydrogens. And this hydrogen's kind of attracted to that oxygen. And so that's how you kind of get water, how it kind of sticks together. That cohesion is caused by hydrogen bonds. Now these are relatively weak. They're enough to play a role in very small things such as proteins and how they kind of form together. In dyes, they're not quite as important though. Next bond is called covalent. And you can think of this by kind of destructuring that word. Co meaning together, and valent referring to valences or electrons. So this is between things that actually share electrons. So a lot of organic molecules tend to be covalently bonded. And so let's say in this hexane, all these carbons are sharing these electrons. And it's because those electrons are going back and forth between these that the atoms are kept together. And covalent bonds tend to be very, very strong. The last type is Vander Waals. Vander Waals forces essentially just occur between any two molecules. And it's just because the electromagnetic charge on both of them kind of attracts them to each other. It makes them incredibly weak. It means that anything near anything is somewhat attracted. But it's so weak that it's completely overcome by any other type of bond. Let's talk about the theory behind nuclear staining. Now, there are two ways that a nucleus can be stained. One is by staining with a basic dye, which tends to be positively charged. So it's kind of sitting out here, and because we have DNA and RNA in our nucleus, those tend to be negatively charged. So as you can see, we have a positive and a negative charge, which, as we just talked about, forms an ionic bond, which is fairly strong. And so that's how we get that purple dye, typically some sort of hematoxylin, to stick to our nucleus. Now the other way is by putting the dye on, even though there's no DNA or RNA in the nucleus, which can happen if we do some sort of decalcification. And then we hit that with a metal mordant, which is something that we might get into in a little bit. But essentially that's what makes the dye stick to the nucleus in that case. Next, let's look at our cytoplasmic staining. So cytoplasm is mostly proteins, and proteins tend to have both positive and negative poles, depending on the chemical groups on their heads and their tails. And so that makes them kind of like our nucleus, but also kind of not like our nucleus as far as charges go. So you could imagine if we stained a cell with the hematoxylin and the protein was mostly negative, then it would probably stain the protein too. So everything would be that hematoxylin purple. It wouldn't really be very helpful because then everything's the same color. The whole point of staining is to be able to differentiate between one thing and another. So how do we get this protein to act the way we want it to? Well, if you change the pH, then the protein itself will actually change its charge. So if you change the pH to less than 6, so somewhat acidic, then this protein is going to be mostly positive. And at that point, we can introduce a negatively charged stain, and which is typically Eosin. Eosin is negatively charged. So those opposites attract, just like they did with our Hematoxylin and our Nucleus. And now you have a positively charged protein labeled with a negatively charged Eosin and a negatively charged Nucleus stained with a positively charged Hematoxylin. So we know how to attach our dyes to the things we wanted to attach them to, at least in a basic H&E stain. But what's in the dyes and what makes them act the way they do? Well, we're going to get into that. Those of you that are familiar with organic chemistry will probably recognize our next piece, which is the benzene ring. Now, a lot of our dyes are created from benzene or coal tar. And what makes these special is they are benzenes, but they're modified benzene rings. And the way that they're modified is by adding certain groups that kind of destabilizes the ring. So that might be an NO2 group or something like that. And what that does is it takes benzene, which is typically not able to be seen by us, though it does give off a certain radiation in the UV spectrum. Human eyes can't see it, so we add some things to it to destabilize it even more. And because it's so unstable, all the electrons, as they're going through all these bonds, kind of jumping in and out of different valences, and as they come down a valence, they release some energy. That energy is in visible light. And so basically we just destabilize this molecule so that it will move its electrons around enough to put it into a spectrum that we can see. When we're looking to name these, this group by itself is a chromophore. So chromo, color. And then if you have a benzene derivative that has a whole bunch of these, that contains all these different groups, the whole thing is a chromogen. It turns out that having a chromogen is not enough to stain something. So in addition to our chromogen, we also have to have what's called an oxychrome. And what an oxychrome is essentially an ionizing group. So it's something that kind of acts as a link, an ionic link, between your chromogen and whatever you're trying to stain. So without both of those, you're not going to have your chromogen, which is essentially your dye, sticking to the things you want it to stick to. So without an oxychrome, you could put the dye in with the tissue, but it wouldn't actually stain anything. It would just wash right off. Now, when we're talking about different types of dyes, you might hear the term acidic or basic, and that can be really confusing because we just talked about pH when we were talking about the staining the cytoplasm, and you hear acid and base, and that kind of confuses things. The better way to think of those is a basic dye, because it doesn't actually have anything to do with pH. So a basic dye is one where the dye itself is positive. So that's a positive dye is a basic dye. Or sometimes a cationic dye, which is essentially the same thing. I'm saying that it's positive ion. And now, if you hear an acid dye, that's a negative dye, or an anionic. But essentially, just remember that's a negative. So that would mean that our basic dye is like our hematoxylin, because it is attracted to our negative nucleus, and our acid dyes are our eosin, which is attracted to our positively charged proteins. Now that we know what dyes are and how to attach them during a routine H&E stain, are there any other factors that we have to worry about? Yes. So, as we talked about, pH can affect things. So, changing the pH can change the charge of things. So, if you're trying to ionically bind something, the pH matters. Increasing the temperature will increase the rate of reaction, typically. If you raise it too high, it can actually denature some proteins, which can cause some some strange things to happen in the staining. Either they don't stain, or maybe they stain too much. Having too low of a temperature can cause the reaction to happen too slowly, and possibly to the point where charges change and things aren't set up the way that you need them to stain. Dyes can increase their binding if there's more of them. So having a high concentration of dye can change how quickly or how effectively they stain something. And this matters because a lot of times when you stain something, you want to get rid of some of it. So having the highest concentration of dye isn't always the best thing, because if it stains things that you don't want it to, or stains too intensely that you can't differentiate between one thing or another, then that's not great. A lot of things we talked about were ionic, and so the presence of salts, which are just ionically bound chemicals, can change how things bind. So having salts in there in your solution that's not supposed to be there can really change things. And the other one is kind of complicated, but essentially when you're doing a stain, you can kind of make the dyes compete. So if one dye is sitting on, let's say, this part of the tissue, and then you add some more dye here, unless this dye has a greater affinity than this one, the original dye is just going to stay here, and this dye is just going to pass that spot by. So that's why we have a certain order to how we do our stains, so that we can only stain the things that we want to, and then we don't kind of erase our work with the next stain. Now that we know how to apply our stains, let's talk about how to get rid of the stains that we don't want. So let's make a little cell here. And a layer. Let's say that we wanted to use this purple dye. Now, we don't want the entire cell to be stained like that. We want to get rid of the stuff that we don't want. So we would differentiate, meaning that we would take away all of the stain that we don't want, and only keep the stuff that we do. So how do we make this kind of effect? Well, when we're staining, our stain tends to come with some extra pieces, which are typically called mordants. And what a mordant is it's typically a metal. It acts as a link between the dye and the tissue. So it's kind of like a transfer vehicle. And it also helps to kind of keep it in place when you get rid of all the excess. So we're going to differentiate between the stuff that we want and everything else, because the mordant is kind of keeping it in place. As it turns out, though, if we add extra mordant, well, that mordant's trying to transfer all of the stain. And so essentially some of the stain kind of sticks to it and tends to go outside of wherever it is and sticks over on your mordant, which can then be washed away because it's not actually touching the tissue that it affects. So by adding an excess of mordant, you're giving the dye something to touch that isn't the tissue. And when you go to wash everything away, the stuff that is touching the tissue stays, and the stuff on the outside all gets washed away. So that's one way that we can differentiate a stain. But what happens if we're using a dye that doesn't have a mordant? So something like our Eosin. So what we do there is we add something of kind of the opposite. So if it's a cationic dye, you use an anionic solution to kind of pull it out. So let's say in the case of our hematoxylin, which is positive, right? Then we would add something negative. What that kind of does is the same thing that our mordant did, where this charge that this dye is attracted to kind of pulls it away. Now, granted, we don't want it to pull away from the nucleus, but these are going to be pretty well stuck in there. But you might have a few stragglers sitting out in the cytoplasm, and so that's why these things are here. So that's why I use that solution. Just kind of pull those out of the cytoplasm, and it also keeps them from competing with the eosin that's going to come after that. Our third method of differentiation is where we oxidize something. So let's say we had a piece of dye here that we just didn't want to be there anymore. Well, if we use an oxidizer, then it would take away the stuff. It's pretty selective, so it only really takes away things that aren't already bound. So it's oxidizing this dye. It doesn't necessarily wash it away, but it makes its chromogens inactive, or sometimes destroys them completely. So though the chemical may remain attached to the tissue, it will not have a color. So when it is shown on the slide, you're not going to see it. And lastly, and it's kind of hard to demonstrate this one, you can use a buffer to differentiate things, which is essentially like using pH to decide whether something stays or goes. So you're kind of controlling the charges through the pH, kind of like we did with eosin. But in this case, only the things that will stay at that pH will stay. So you use a buffer to kind of select which dyes will stick and which ones will leave once they enter that buffer. Now let's take a look at some of the origins of our natural dyes, and a little bit about hematoxylin in particular. So hematoxylin is derived from the heartwood of the logwood tree, and that actually became an issue at one point in the past. Logwood became very hard to get a hold of. I think it came close to extinction, so we had to use some different alternatives to hematoxylin in the laboratories, though I wasn't working in a laboratory at the time. Carmine is an important natural dye, and that is made from the shells or the carapaces of cochineal insects. Orosin is derived from lichens, and saffron is made from the flowering saffron plant. And so some of those are the natural dyes that you might encounter in a histology lab. Now, with hematoxylin in particular, you're going to see some terms that seem similar, but they're not. So when you look at something like hematin, which is what we're really talking about when we talk about hematoxylin, is the oxidized thing that comes out of the logwood tree. So that's our dye, right? So that's hematin. Hematin, on the other hand, is something you also see in histology circles, but that's referring to things like formalin pigment. So it's not even closely related to hematoxylin. So hematin has 4 syllables, so that's the thing you're looking for. The hematin with the 3 syllables is more in pigments and things like that. So it's important to keep the two differentiated. Now let's talk about hematoxylin for a second. Hematoxylin has to be oxidized in order to do its thing, in order to act as a dye. But if you oxidize it too much, it starts to break down. So we've devised different ways of reaching that perfect oxidation point, and that has led us to make different types of hematoxylin for different uses. Now, some hematoxylins are ripened or oxidized to that perfect point naturally. So you just kind of keep it out in the open air, and the atmospheric conditions will oxidize it. Sometimes we'll use a chemical oxidizer, and all this really depends on what else you have in there, and that determines what is the best way to oxidize your hematoxylin. The first hematoxylin I'd like to go over is Harris hematoxylin. This one's pretty popular. You're going to see this a lot on routine stainers and that sort of thing. So what's in Harris hematoxylin? We have our hematoxylin, so it's our raw dye there. We have some absolute ethanol, and then ammonium aluminum sulfate, some distilled water, and sodium iodate. And this used to use mercuric oxide, but for obvious reasons we're not using that anymore. We're using sodium iodate. So what does all this do? Well, our hematoxylin is obviously our dye. The absolute ethanol is just a means of kind of dissipating that dye and sort of leaching the dye out of the raw product. Our ammonium aluminum sulfate contains aluminum, and that's the thing we kind of want to look at there, because our aluminum acts as our mordant. So as you remember from before, our mordants are kind of a vehicle, a way of transporting that dye and creating a link between the dye and the tissue. So that's what our aluminum is doing there. And the ammonium and the sulfate are just ways of making it non-metallic. And so that allows it to be more reactive in the way that we want it to be. Now the sodium iodate is our chemical ripening agent. So that means if you're keeping a list of hematoxins that are ripened chemically or ripened naturally, Harris hematoxylin is going to be in your chemically ripened list. There's one other thing that you can add to Harris hematoxylin. You can add a little bit of acid to it. So some folks will recommend a 1% hydrochloric, and that's to essentially get the right amount of differentiation. See, Harris hematoxylin originally was meant to be as a progressive stain, and what that means is that you want to put the slides in the stain just long enough for the stain to get to where you want it to be, and then you take it out. So you let it stain and then stop the process when it reaches the point that you want it to be at. And that can be compared to regressive staining, where you intentionally overstain the slide and then differentiate it back. So kind of put too much dye on it and then take away some of it. And this just kind of depends on what kind of dye or stain that you're using will determine which of those techniques you want to go with. Speaking of regressive staining, Delafield is one that you would want to put on the regressive list. So to make Delafield, you have two solutions that you're going to put together. First is your ammonium aluminum sulfate, so that's your mordant. And the second is the hematoxylin and some 95% ethanol. And then once those are mixed together, you're going to ripen them naturally. So Delafield is also one of our naturally ripening hematoxylins. So after it's ripened, then we'll add our glycerol and our 95% ethanol. Now the use for the glycerol is to stabilize this solution over time. So in this case, the ethanol acts not only to get the dye out of the raw hematoxylin, but it's also meant to act as a preservative. So that kind of keeps things from decaying. So when you're naturally ripening hematoxylin, you have to have a way of determining whether it's ready or not, because there's really no set way of doing that. You're just kind of letting the atmosphere do its thing. So you kind of have to test it along the way. Now you can make a general guess after making a couple of batches to figure out how long it might take, but you still have to check the endpoint. Now luckily, hematoxylin tends to have a good point, so it goes from being kind of more raw to when it's ripe, and when it's overripe, it goes back to the kind of the same color it does when it's raw. So our good hematoxylin is going to have like that nice deep purple color, but then before that, it might have kind of like a reddish hint to it there, and then when it's over oxidized, it goes right back to it. And so when you're looking to see if things are ready or not, you might even have a little bit of this reddish stuff in the middle if you drop it on some filter paper, but you want to make sure it has a ring of a nice deep purple on the outside. So when you're checking to make sure that it's ripe, overall, you want it to be this nice purple color. When you drop on some filter paper, it'll probably be this red in the middle with a halo of that purple on the outside. Also, you can kind of tell by the smell whether hematoxylin is ready or not. It should smell kind of like wine. If you have alcohol in the solution as your counterstain, you want to have something that doesn't have alcohol in it, because the alcohol will actually dissolve that chromogen. So mirror hematoxylin is recommended specifically for that technique, but also mirror hematoxylin doesn't have the scum on the top because the pH is kept at the correct level. So other hematoxylins go ahead and kind of get a little bit of a scum on top of the solution. So Mayer tends to be pretty stable as well. So you might see it in some special stain techniques in your laboratory, just because you can keep a bottle of it for a pretty long time, it'll stay stable. Our next type of hematoxylin is Mayer hematoxylin. And so this is one of our progressive stains. So let's see what all is in it. We have our hematoxylin. We have some distilled water, our sodium iodate, which we've seen before. That's going to be our chemical ripening agent. So this one is chemically ripened. We have our mordant, which is our potassium aluminum sulfate or ammonium aluminum sulfate. Then we have citric acid and chlorohydrate. Now notice that there's no alcohol in Mayer hematoxylin. And the reason for that is citric acid and chlorohydrate kind of do some of the stabilizing and kind of get the dye out of the raw product there. But also, they keep the pH at a certain level. And so between keeping the pH at a certain level and also not containing alcohol, that makes it kind of special, because when you're using something like an aminoperoxidase, and you're specifically when you're using a 3-amino 9-ethyl carbazole, next hematoxylin is our Ehrlich hematoxylin. What's inside of that? Well, we have a regular hematoxylin, some ethanol, some water, glycerol for stabilization. Our mordant is ammonium or potassium aluminum sulfate. And we have some glacial acetic acid, which we said in some of the other hematoxylins you can add some sort of acid to kind of give better differentiation or sharper detail. And Ehrlich is essentially just one where you can do that in a big quantity and just kind of do that all at once. Now notice that we don't have a ripening agent included in this, so unless you make it into a chemically ripening hematoxylin, that is going to be natural. Though the text does say that you can add some sodium iodate to speed up the ripening process, so it's a little bit more flexible in that way. And this one tends to be used regressively, but it can be used progressively. So Ehrlich is the reason why you might use Ehrlich is it's pretty flexible, and it tends to perform fairly well and gives that sharp detail. Next, we have Gill hematoxylin. Gill hematoxylin contains some distilled water and ethylene glycol, which in this case is going to be used instead of alcohol, like we've seen in other hematoxylins, and that's essentially going to do the same thing. It's going to kind of pull that raw product out or act as a solvent for the hematoxylin. In this case, it's very specific about using anhydrous hematoxylin with the formula given, and that's just because if you use crystallized hematoxylin, then you just have to use different amounts. But really, the important thing is that you just have some sort of hematoxylin in there. We have sodium iodate, so this is chemically ripened, though there's a caveat with Gill that you can use it immediately, but it does work a little bit better if you give it a few weeks to ripen. You have aluminum sulfate as our mordant, so we have our aluminum in there. And then we have glacial acetic acid, which acts as our acid component, which gives it a bit more of a sharper contrast, a little bit more detail. So Gill is pretty interesting in that Gill can be used at different strengths. So as you add different amounts of hematoxylin, the staining quality gets more intense. So if you're buying Gill hematoxylin, then you might see it in different strengths. So Gill 1, Gill 2, or Gill 3. So things like Gill 1 and 2 can be used progressively and just won't overstain at all. So that's pretty handy if you have something that maybe you're staining something and you can't pay a lot of attention to it. Those are pretty safe to use. Gill 3 might overstain. Something else to know about Gill is it will stain goblet cells. So if you're using Gill on routine H&Es, just know that those goblet cells are going to be stained. So that if you might be looking into something where you need those to be not purple, then maybe Gill isn't the stain for you. Something else that you might see associated with hematoxylin is a bluing solution. In this case, we're looking at Scott solution, which is fairly common. In Scott solution, you have magnesium sulfate, sodium bicarbonate, and water. Now, the whole purpose of Scott solution is to blue our hematoxylin. And what bluing is it's a process where we change the pH to something above pH of 5, and the hematoxylin changes color from kind of that reddish purple to a more vibrant purple. and also it solidifies the dye. So up until this point, we've kind of relied on our mordant to kind of keep things together, but once the pH changes, that dye really sticks in place. So it's very hard to get it off of there after that, which is important because typically we're going to stain our slides with hematoxylin first, and then we're going to add something else on top of them in a routine stainer, so typically Eosin. So that hematoxylin has to stay in place during that whole process, and bluing is essential to that. Our last hematoxylin stain is Weigert, and the reason we have this after bluing and everything else is because it's fairly special, and it's not really used in routine staining. It tends to be used in special staining. And what makes it special is the ferric chloride, for the most part. So you have your Weigert comes in two parts. Your ferric chloride, distilled water, hydrochloric acid are in solution A. And then in solution B, you have your hematoxylin and your ethanol. And so you just mix those together and you have Weigerts. Chloride is special because it has iron in it. And so that makes this an iron hematoxylin as opposed to an aluminum hematoxylin. And that difference makes it much more resistant to decolorization. And also this ferric chloride not only acts as the mordant, but it's also the oxidizer. So it's ripe and ready to go right when you make it. Though you want to keep the hematoxylin separate so it doesn't over-ripen. So that's why we have it in two different solutions. And we also have our hydrochloric here for that better detail. As I mentioned before, the hematoxylin is derived from the heartwood of the logwood tree. And so that makes it kind of a naturally occurring and somewhat limited resource. And at one point in the 1970s and in 2008, actually, we had shortages. And so we had to rely on a few alternatives. One of those alternatives for nuclear staining was Celestion Blue. And Celestion Blue contains Celestion Blue, ferric ammonium sulfate, and distilled water. It can be produced synthetically, so we can make it in huge amounts. My guess is it's probably just more expensive, and that's why we don't continue to use it. So we have a substitute for our aluminum hematoxylin if we run out of hematoxylin. So what do we do with our iron hematoxylin stains or Weigerts? Well, we can use Galine as a substitute. So in that case, we make it very similarly to our Weigert hematoxylin, where we have two different solutions. Solution A has our Galine, our actual dye, ethylene glycol and some ethanol, and in solution B we have some water, hydrochloric acid, ferric ammonium sulfate. And so you just add those together and you'll get kind of something that looks a lot like Weigert hematoxylin. And Galine, from what I can tell, is just made synthetically. So I honestly have not heard a lot about this since studying years ago. So my guess is it's not used very often outside of a shortage. Now, when we're talking about plasma staining, typically you're going to use some sort of Eosin. You might use Eosin Y or Eosin B. Most of our Eosins are going to contain alcohol, glacial acetic acid, and the pH is going to be around 4.6 to 5. Some folks do a Phylloxene B Eosin, and that can be helpful for making more of an intense Eosin stain, but it's also easy to overstain with that. And so when you are using your Eosin stain, typically you want to have at least 3 shades. So you should see red blood cells are going to be kind of this deeper red. Things like collagen, so around any kind of fibers or things like that, should be a little bit of a deeper, like dark pink. And the rest of the cytoplasm should be this almost bubblegum pink. It should be fairly diffuse as well. So when you're making control slides to check your H&E stainer, you want to have something that has some nuclei, which is not hard to find, but also you want something that has collagen, red blood cells, and obviously cytoplasm, so that you can make sure that all of your stains are working correctly. So I was just about to get into the part where we start kind of drawing together, and I realized that my touchscreen is now malfunctioning. So I'm just going to ask you to bear with me here. The screen may have changed a little bit. I'm sorry if it makes it a little bit blurry, but that's going to give us the most area to draw in. So if you'd like, you can follow along with me. If not, you can skip through some of this stuff as I kind of take people through the drawing process. I feel like if you're an interactive learner, then this is a great way to kind of get in there and make this a little bit more real for you. So you're not just observing and listening, you're actually taking part in this process. So we're going to go through the manual H&E staining. Now, if you'd like to skip ahead, go ahead and skip until you see a picture of some tissue without any color on it, but some gray shadow over top of that. But for the rest of you, if you just want to follow along, well, I'll take you through this. In order to show all of the things that we would see in an H&E stain, a lot of times you'll pick a control tissue that includes all of our different things that would stain with eosin. So that would have collagen, some erythrocytes, red blood cells, and obviously cytoplasm and nuclei. For me, from things that I've seen, gallbladder could be a pretty interesting control and fairly useful because it has some epithelium, but it also has some arteries and some muscle tissue, and so you're going to get all three of those shades, and they tend to be kind of organized in a way that's easier to tell if your stain's working or not. Now, I'm not sure what everybody uses for their controls, but we're going to kind of draw a piece of gallbladder and go through the staining process with that. So, first thing I'm going to do is add a new layer down the right side there. You can do all these things however you do with your graphic editor. I'm using Krita. So if you're using Krita, you can follow right along with me. I'm just going to kind of draw the outline here because we're going to start from the very beginning. So let's say something came off the microtome. We're going to do our little outline, even though sometimes these outlines are not all that visible. You'll usually see the outside of it. Now that we have our outline, let's keep in mind that this just came off of the microtome. So it's going to have some paraffin over it. So I just made a new layer and picked up an airbrush and set it to gray, which just kind of makes sense for this. I'm going to set that at a low opacity so we can kind of still see through it. That's just to illustrate that this is indeed there. So we have our paraffin over top. Okay. There we go. Now, as we go through our staining, obviously we're going to try to get rid of this paraffin. It's going to be our first thing, because that's kind of our first barrier to getting anything on this tissue is all this paraffin. So how do we want to get rid of that? Well, in staining, typically we are going to use xylene or a xylene substitute. But we're just going to go with xylene here. If your lab doesn't use that, just know that you'll use the xylene substitute in these steps. So the slide has just gone off the microtome, and maybe it's gone onto the heater, which has melted the paraffin, but it doesn't remove it. It just kind of makes it kind of start to break down and be a little bit more susceptible to the xylene. It also kind of fixes the tissue to the slide a little bit, but for the most part, that's just to kind of break down the paraffin there. So we're going to hit this slide with some xylene, and so that's going to kind of erase some of this. So I have the eraser selected. So this is just getting a little bit lighter here. Alright, and then we're going to just hit it again. So we're probably going to dunk in xylene like 2-3 times. As you can see, it's just getting a little bit lighter as we go. Okay, cool. Now, this is not a blank slide, of course. We just add a bunch of xylene to it. So we're just going to use blue for that. Kind of a light blue. We're just going to keep that opacity small so we can still see the background here. But we have some blue to indicate our xylene. Because now we have xylene on our slide, and we can't really stain anything while there's xylene on it. So what do we do with that? Well, we're going to add some sort of an intermediary, right? We're going to add something that we can wash off and get to the stain. So we're going to put it into some alcohols. So we're going to use green for that just to signify it's a different thing. So it might be 1, 2, 3 changes of alcohol. Okay, now we're covered in alcohol. The next part that we typically use is water. Now water is different than any of those other two reagents because it does kind of reset things to a point where we can actually kind of see things. So we're just going to use the eraser on this part. Alright, and you got to put it in water for a while. I mean, it really does take a long time. But I am going to increase my opacity here because this really does get rid of all of it eventually. Okay, great. So we started out with a piece of tissue that has a whole bunch of paraffin on it, and we've successfully deparaffinized it with our xylene, our alcohol, and then our water. And now that it has water on it, it's essentially open. It is open to whatever stains we put on it. Let's go ahead and select a color for our Hematoxylin. It's going to be a fairly deep purple color, even a little bit towards the reddish side, just because before it's blued, it does kind of keep that wine color, and then it's going to be close to a purple later on. So it could even be like a dark wine, or even around this. That's a pretty good one. So, because we're putting the whole slide in, now we have Hematoxylin on everything, because we're about to hit our muscle layer, our smooth muscle layer. So I'm just going to bring the size down to about, it might actually be too little. Yeah, it's way too small. Well, let's try a little bit bigger. There we go. And we just kind of want to outline the things that are going to be kept. Okay, then we get up into our epithelium, same thing. And how does this work on a chemical level? You know, when you're not actually drawing your dyes on, how does that work? Well, if you remember, we are kind of relying on the negative charge of the DNA in these nuclei to keep this dye right where we want it to be. So we're just kind of circling everything. That indicates that the dye is fast, meaning that it is kind of steadfast. So when we go to wash off the excess, it's already stuck there. And it's stuck there because of our mordant. So in this case, if it's Hematoxylin, that is going to be because of the aluminum. Okay, then we can kind of use a bigger eraser here. Let's get the rest of this done. And we can actually bring the opacity up pretty high, because this is going to do a pretty good job. Not a great job, pretty good job, right around the cytoplasm here. And the reason for that is because we are going to accentuate this just a little bit in a second. Okay, so yeah, we're just going to get rid of all this extra stuff on the outside. The majority of it, once again, not all of it yet. But it's going to get rid of quite a bit of that extra Hematoxylin. Now, let's say that we did not have an acidified Hematoxylin. So the next step would be to add that acid to it. So then we're going to use a slightly smaller eraser just to get around all of those little bubbles that we just made. We're going to use a pretty high opacity here because this should get rid of everything. And don't be afraid to kind of just cut right through those. Oftentimes you're not going to see the cell membrane. So yeah, just going to kind of cut through these. Trying to cut through the nucleus like I just did. There we go. Okay. So yes, this acid is just accentuating these and it's making them a little bit more crisp. If you want, you can actually go back and kind of fill these back in. This is more just to kind of visualize what our acid is doing in this case. In fact, I'm even going to do that. So we're just going to go ahead and color pick that and turn off our eraser and just kind of refill in those nuclei. Because yeah, the acid really does help make these show up a lot better. And this isn't going to be picture perfect. And if you've ever seen an H&E slide, you know that that is always the case. That you always get a little bit of noise in your stain. And that's okay, as long as it is able to be read by your pathologist, you're good to go. Also, these aren't all that easy to read without a counter stain, which is part of the reason why we use a counter stain, but also to stain our collagen, our erythrocytes, and our cytoplasm, all those elements. But you will find in some of our special stains that those counter stains are almost completely to add some sort of contrast to make it easier to see these other components. So far, we have gotten rid of our paraffin, we've washed everything out, all the reagents, and then we've added hematoxylin, and then we've accentuated the nuclei with an acid. Now there's one more step that is tied to hematoxylin, because we still have kind of this wine color here, and that is to go through bluing. So you might use a Scott solution, you might just use something that is fairly basic, just to get that pH around 8.5 is usually pretty good. And I'm going to use a different brush because these are very well defined once you put them through the bluing. And so we're just going to color right over those. Remember, as we're drawing these, or maybe as you're watching me draw these, that's essentially what we're doing with our stains. Because you see, when the pathologists are looking at these slides, they're not really looking at the components themselves. They're looking at the dyes that we're painting the components with. So that's why our job is so important, is because without these dyes in the right place, things could look like things that they're not, or without any dyes at all, without us here to kind of paint these pictures for our pathologists, then they'd be looking at an unstained slide, in which case you can't really see anything at all. Okay, so we've blued everything. Great. Everything is done on the hematoxylin side. We'll probably wash everything off. So I'm just going to get our eraser. And I'm just going to go with the airbrush again. Except it needs to be ready to erase. There we go. A little bit bigger just to make it a little bit easier. And there we go. Now notice, because we made a new layer there, you don't have to worry about it really hurting the outline. That's why we do things in layers. And it's also why we stain in steps. So if we threw everything, all of our stains, all in at the same time, what would happen is you would have stains competing with each other. So the hematoxylin would be competing with the eosin, and the pH would be weird, and you would just kind of have this weird amalgam of dyes, and you wouldn't get all the detail that we're getting right now. And so once we wash all that off, because we have everything stained that's going to stain with hematoxylin, we can simulate that competition. The fact that the hematoxylin is there and nothing else can attach unless it has a greater affinity, which we're not going to use anything that has that. So we're going to simulate that by creating a new layer. So this new layer is going to go on top of everything we just did, much like when we stain. So anything that comes after hematoxylin is just going on top of the rest of the tissue that's not hematoxylin. So our next step, we're going to get into our eosin. And what I would do for this is go with our very fine pieces first, or our more heavily stained things. And the reason for that is not only to make it a little bit easier to draw, but it's also kind of the process too. So if you figure, if something is staining more deeply with eosin, so it's getting kind of a deeper red, so that's your erythrocytes, or typically around arteries that have a lot of erythrocytes, then that's because it has a higher affinity for eosin. So it's going to pull those in first. And that's why it gets that deep red, is because there's a higher concentration of eosin in that spot. So what we can do here is we can kind of mess around right below the smooth muscle layer, but still above the serosa. So right around the connective tissue, we can put a few arteries in here. Once I turn off my eraser. There we go. So yeah, we'll put a few arteries here. And honestly, these are pretty accurate as far as what you would see on a section. They are very, very deep red and tend to have some odd shapes to them depending on what angle you cut the artery at, or whatever blood vessel it happens to be. And then next, we're going to move on to our collagen, which you're going to find a lot of in muscle. It tends to be a little bit of a softer shade, and a little bit of a softer shape even. So we're just going to kind of get in there. It's a little bit too hard for me. So we're going to make this a smaller size for our airbrush. And there we go. And I'm going to bring the opacity way down because it is fairly faint. And we might have maybe a few too many nuclei in this spot. That's okay. And then remember, when we're drawing these, you don't have to worry about necessarily messing up the nuclei underneath, because those are in different layers. So when we draw on top of them, it's not going to necessarily smudge them up, but it is going to overlap them. So don't draw right on top of your nuclei if you can help it. And we'll just kind of put some more collagen fibers there. And that's our smooth muscle layer, which we should see if we're looking at a gallbladder. So after that, we're going to get to our last layer, which is right above the serosa, but underneath our smooth muscle. And we're going to go with just a slightly bigger airbrush, because this is our other shade of eosin. So this is kind of that bubblegum pink you're going to see on everything else. So just kind of color everything in with that. It's okay at this point if you kind of cover everything, because eosin has to be differentiated just like our hematoxylin did. So we're going to be getting in here with an eraser relatively soon. Okay, so now our tissue is completely covered in eosin. And eosin is alcohol-based, so this isn't going to get a wash. It's going to go right into a graded alcohol typically. So you might put it into 95, you might put it into a lower concentration. My laboratory in particular puts it into 1 change of 95 and 3 changes of 100. So I'm just going to switch to our eraser, because differentiation just erases that layer of eosin. So we're just going to kind of get rid of the outside. So we're going to need a much smaller eraser to get rid of the rest of it. Okay, great. So you call this maybe your 95. Might look a little something like this. I'm just going to kind of get it in there. Okay. So that'd be our 95. Now we'll go through our hundreds. I'm going to make the opacity at 100, and I'm going to make the size much smaller, because this is going to be some very, very fine differentiating. So we're just going to kind of get in there. So inside of our blood vessels, it's actually going to be completely clear, because there's no cytoplasm in there. That's actually a tube by itself, where some of the red blood vessels are going to hang out, but it's not going to be so clogged with them that there is a solid spot. So you're going to see some white space in there when you have red blood vessels. And we're going to take away our eosin from our nuclei. Our 100s help with that. Okay, so just kind of uncover your nuclei a little bit. Now, if this picture in particular was shown to a pathologist, they might say that these nuclei are a little smudgy, and that would be a fair evaluation because we are doing this with our people hands, and people hands are not nearly as accurate as the chemicals that we use. And honestly, that's just a testament to our procedure. Because really, I don't know, if we could paint these by hand, would we? No, we still wouldn't. Even if it was more efficient, it is so much more accurate for us to use our procedures that we have in place. So we're just going to continue uncovering all of our nuclei there. We might have lost a couple of our shades here, so we're just going to paint those back on. And that one's a little bit too dark. Just going to go with that. There we go. And maybe just a little bit lower opacity, because this is a small diameter to use for our airbrush. So that makes it a little bit thicker. Now I'm just going to pick this one back up. We lost an artery back there. We don't want that. Okay. So you kind of get the picture there. So we have our tissue. So we have kind of our control tissue here. And like I said, it's a little bit smudgy. You wouldn't probably want to hand this in for a doctor to look at, but you get the idea. So when we go to stain something, we have to immerse everything in the stain and get rid of what we don't want. And whether that's through washing it or using some sort of chemical agent to differentiate it, that's how we get more than one color and get those colors on the things that we want them to be on. So while we're here, just a few troubleshooting tips. For one, if your nuclei are as smudgy as we've made them here, so it's really smudgy like that, you might have some sort of a fixation problem. So maybe they're underfixed, or maybe have some autolysis going on there. So they shouldn't look this smudgy. So they should look a bit more crisp. Maybe something along the lines of this one. It's pretty good. Now let's say that you came across a bunch of white marks. So just clear spots where there should be staining, but there isn't. So if it's like this, and it's in the whole staining, and you're just kind of wondering, well, where did those come from? Well, if you remember, our first step was getting rid of paraffin, right? Because paraffin tends to block our stain. And so if you don't get rid of all of your paraffin, you can kind of get a kind of like a white area where just there's nothing there. There's nothing is staining correctly. And so if you're finding white spots, just make sure that your deparaffin step is running well. Now sometimes, if your colors are off, which this Eosin isn't great, the cytoplasmic Eosin in this example is a little bit dark now that we've colored over it a lot. So if it's that dark, then you might want to check your Eosin. It should be more of that bubblegum pink color. But also, if your Hemotoxylin is a little bit pale, it could be that the Hemotoxylin is too old, might need replaced. Now, if your nuclei are actually too dark, which that can happen, so let's say instead of being this kind of purple color, you're getting something like this. kind of like almost like a blackish color there, and won't have any kind of transparency to it, chances are you have a thick section. At that point, you might want to check your microtome. Maybe it's cutting thick and thin, or perhaps it's just set at too high of a thickness and you're getting 2 layers of tissue instead of 1, and so you're getting twice hematoxylin on the same spot. If your nuclei still have that kind of reddish brown or that wine color to them, then chances are they didn't blue right. Because remember, they were that color before we blued them. So maybe your bluing's not working. You might want to check the pH of it, or maybe see how old it is. It could be that it just needs replaced. Now, let's say we really just want to see our nucleic acids. So instead of using a hematoxylin or an eosin, maybe we just want to see where our DNA and our RNA are. In that case, we might want to use just a nucleic acid stain. So our first one of those is a Falcon reaction. So in a Falcon reaction, what we're going to do is acidify the tissue. And what that does is it kind of breaks the DNA down a little bit, and it exposes a part of the DNA that has some sugar-sensitive phosphates. So it's kind of getting rid of all of our adenine, guanine, cytosine, thymine, and just leaves behind that backbone, that sugar and phosphate backbone. And with that being exposed, we can attack that with a Schiff reagent. And what a Schiff reagent does is it reacts with aldehydes, which are sugars are aldehydes. So the Schiff reagent attacks that, and then that will then demonstrate the DNA. And then we'll use a counterstain to essentially accentuate the rest of the tissue. So let's say we did this on a lymph node, because the reason I like lymph node for Falcon reaction is because in the nodule you have a ton of nuclei all in one spot. And then you can see on the outside of the nodule you have all the connective tissue. So you should have that kind of purplish color. Maybe it can be a little bit darker, a little bit lighter, depending on your shifts and how long you do this. But on the outside, you're going to have a clear distinction where that color should stop. So if we wanted to draw this, I would start by just outlining our nodule there, and maybe just give it some connective tissue on the outside. Then I would make a new layer. So this is a layer that is not going to affect our outline here. And now some of these steps aren't going to have any color associated with them, but we're going to go through them anyway. So your first thing you're going to do is rinse your slides in some cold hydrochloric acid. That's after deparaffинizing, obviously. We're just going to kind of take that for granted at this point. Then anytime you want to stain something, you need to deparaffinize it first. So after that, we rinse our slides in some hydrochloric acid, then we put some in some preheated hydrochloric acid, and then we're going to rinse it one more time in cold hydrochloric acid. And all that's doing is it's allowing us to kind of break that DNA down and remove all of your base pairs, so all of your actual nucleotides, and just leaving that backbone, that sugar phosphate backbone, to be attached by the shifts. So we've already put through all of our acids. Then we're going to stain it in shifts for about an hour. Then we're going to wash our slides. So shifts is kind of funny in that it can kind of start to stain while it's in the shifts, and then after you wash it, it tends to get all the rest of the intensity later. So we're just going to paint that as if we've put it in our shifts and we've washed it already. So see how all of our nuclei are right around in the nodule. Now the outside is relatively clear. And granted, if you give that to a pathologist, they could see that there's DNA there. And that could be the end of it in a lymph node, but maybe it's something else where the DNA is more sparse. Then the counterstain is a lot more important. So let's say that all of our Schiff's reaction is done, and we've already washed them, so we have our nuclei stained. Then we're going to rinse that in 3 changes of sulfurous acid, and then we'll wash all that off. And lastly, we're going to add our aqueous light green. And so that's going to be kind of this kind of light, well, essentially just light green is kind of what it sounds like. So that'll get kind of everything else, everything that's not, it doesn't have DNA in it. So all of your connective tissue right around your nodule here. Then you can kind of take the size down off of your hairbrush to kind of add all of your connective tissue fibers there, make it a little bit more realistic. And there you go. That's your Falcon reaction for DNA. Now let's say that you wanted to tell the difference between DNA and RNA. So maybe something that has a lot of plasma cells or immunoblasts. Well, you would use a methyl green or a methyl green pyridine Y stain. So in this case, the DNA is going to stain that methyl green, while the RNA is going to be red because of the pyridine Y. And in order to demonstrate this, you might look at something that's lymphoid, some lymphoid tissue. You're just going to have kind of the variety of cells that are going to contain different amounts of RNA and DNA. So if we wanted to draw that, we might just do just a few circles because lymphoid tissue is not always super organized. So just kind of a few circles there, and that'll be our kind of drawing layer. So when we're doing this stain, there's a purification process that comes into using a methyl green. So methyl green needs an acetate buffer to kind of stay stable. So in order to make that acetate buffer, you would take glacial acetic acid and add that to sodium acetate trihydrate in some water. And you kind of put the methyl green into that acetate buffer to keep it stable. And after that's all done, you want to take that and put it into a separation flask or separatory funnel. And you want to add some chloroform to it. Essentially, the chloroform is going to take out any of the methyl violet that is in with your methyl green. So you're trying to purify that green. And so in the separatory funnel, when you add the chloroform, the methyl green violet is going to go into the chloroform in the bottom of the separatory funnel. So then you just keep adding chloroform and separating off the chloroform with the violet. You just kind of keep doing that until the chloroform is then clear. So after it's clear, you kind of siphon off all of the chloroform and let it sit about a few hours, and then you want to make sure that all of that's out, and then you let it sit overnight, and the chloroform will then evaporate off, and you have some stable methyl green with a buffer, and that's good for a few months. So after all that's done, then we want to add our pyridine Y, which is our other part of the stain, and just add those together, and you have your staining solution. So we have our tissue, we deparaffinize it like normal, and we have it down to water. Then we want to stain these with our methyl green pyridine Y solution, and then we want to rinse those off. And then we dip the slides in a couple of changes of acetone, and then we want to go from like acetone to acetone xylene, and then from acetone xylene to xylene. So it's kind of moving it into xylene without having it hit alcohol, which might not be great for our stain. So this stain is different than a lot of them, where you're not necessarily adding one part and washing it off and then adding the second part. You're kind of adding it all together because the methyl green pyridine tends to hit its two targets differently. So the DNA will attach to the methyl green, and the RNA is going to attach to the pyridine Y. So we kind of draw this all at the same time. So we can go ahead and get an airbrush and get our DNA. So we're trying to find that good. Almost a blue in this case for methyl green, oddly enough. Yeah, it looks fairly blue. Okay, so if we wanted to see all these, you might even use a different tool just to make them a little more prominent, because they do tend to have some concentrations of DNA in some of them. Okay, so these are our cells that have all the DNA in them. And our RNA is going to be shown in kind of a rose to almost like a darker red color. So let's go with that. There we go. You can go with that. And this is a bit more diffuse. So you might see, as compared to the methyl green, the pyronin, this will be a little bit more diffusely stained on there. Some of the stains that we use are referred to as polychromatic stains. These polychromatic stains tend to kind of make different colors on their own, and depending on what they come into contact with, which can be really nifty if you're trying to differentiate between two different types of cells. If those cells react with a dye and can be made into different colors, you kind of skip the step where you're painting in layers, and instead you're just painting depending on the reaction of the cell with the stain itself. And a subset of those, a very popular type, is called a Romanowski type stain. And a Romanowski type stain typically contains a methylene blue and an eosin. And Gypsa is a subtype of Romanowski stain. So if you're Gypsa and Romanowski can sometimes be used interchangeably. So if you hear one, you typically want to think of the other as well. So if we wanted to do a Gypsa stain, let's say particularly a May-Grunwald Gypsa stain, much like our last stain, the Gypsa stain, because it's polychromatic, kind of happens all at once, even though this stain does have a couple of steps where we're going to add our Jenner dye, and then we're going to add our Gypsa. The staining itself is really kind of relies on the tissue type, in this case the leukocytes, and where they are in development. So leukocytes in different types of development will be either kind of a pink or a gray or sometimes even blue, which kind of makes them blend in with the other nuclei in the slide. So let's go ahead and just draw a few cells here. And we're going to add some squiggly guys here because we are going to see, actually there might be more that, because there's one more thing that Gypsa can be used for. That is for H. pylori. Typically, it can be used for bacteria in general, but a lot of places will use this for H. pylori testing as well. So, if we go through the procedure here, even though it's not going to be able to draw it really in sequence, let's just go through that. Typically, Zenker is preferred for a fixative if you're going to run a Gypsa. So maybe you're going to run this on a bone marrow or something like that. You might want to put this in Zenker or a B5 first. Then you want to put them in some methyl alcohol. A couple of changes actually. And then we want to use your Jenner solution, which you've made up previously. So your Jenner solution is made up of your Jenner dye and some methyl alcohol. And you want to put that into some distilled water. So go ahead and put that in your Jenner, and then go completely right from the Jenner directly into the Gypsa. And this is pretty typical for any kind of Gypsa stain, is there's not really any washing in between because you're looking for that polychromatic effect where the stains are kind of working together to differentiate the things for you. So you're not doing a lot of manual differentiation in between the dyes. So you typically go from one right into the other. So after it's in the Gypsa for a while, you rinse that in distilled water, and then you differentiate the whole stain with some 1% acetic. And you just kind of, it's not a hard differentiation to make, but differentiating it does get rid of some of the background staining. And so the results of that are going to look a little bit like this. So I'm going to use the airbrush here, and you're going to get some nice, well, definitely not nice and dark. It's actually going to be some nice blues here. In fact, even more of an intense blue, maybe with a higher opacity. There we go. Yeah, so a lot of your nuclei, and sometimes the entire cell is same blue. And then maybe if you have a lymphocyte over here, that's going to be some sort of shade of pink or rose, maybe kind of a grayish rose over here. So once again, that depends on where it's at in development. And then if you had your E. coli, those are also going to be blue. So we're going to kind of narrow down our brush here a little bit. Maybe not that much. And depending on what angle you're cutting these at, they're going to look a little different. But yeah, you'll see the E. coli kind of hanging out. And obviously, you're not necessarily going to see all of these all on the same slide, but we're just kind of color them all at once to save a little bit of time. So yeah, that is our Giemsa stain. That's a May Grunwald specifically. Now let's say that we've completed our stain, everything looks good, we checked under the scope, and we want the pathologist to be able to look at it. Well, typically you want to have a cover slip over that slide, because our cover slip is going to protect not only the tissue from physical abrasions and things like that, but it's also going to kind of seal that stain and keep it from degrading just from contact with the air. So when we put a cover slip on, we usually have to put some sort of resin to bind it. And as we know, when we add things to a stain, that can kind of cloud the things that are underneath. So when we were drawing things, anything that we were drawing on top of kind of got written over. So it's very important that our resinous media or our aqueous mounting media, whichever one we choose, is transparent. And that has something to do with the sort of refractive index. And so what that essentially is it determines how light passes through it. And so in order to make it so that our media and our cover slip are transparent, we want that refractive index to be about the same as the tissue. We want the light to act the same when it hits the media as it does when it hits the tissue. And that way it's almost as if the media isn't even there. And so that is the reason why we might pick a resinous media. So resinous media is essentially, it's synthetic, and it's typically something that is dissolved in some sort of solvent. And so once that solvent dries, then it becomes permanent. It kind of crystallizes in place. And the good thing about that resinous media is the refractive index is very close to that of tissue. So you want something that's between 1.51 and 1.55 in terms of refractive index. And resinous media tends to be 1.53 to 1.54, so it's right there in that nice spot. But the thing with resinous media is you typically have to do some sort of dehydration and clearing to use it. So you have to have kind of a solvent ready on the slide. And some of our stains can't use that. So in that case, you would use an aqueous mounting media, meaning it's somewhat water-based. So you don't have any kind of alcohol or xylene there or any kind of clearing agent present. In which case, yeah, go ahead and use an aqueous mounting media. The downside, why don't we use aqueous mounting media all the time? That would preserve a lot of our stains, is because the refractive index is 1.41 to 1.43. So it's not as clear as a resinous mounting media. So I would say only use aqueous mounting media if you absolutely have to. Now, once your media is on the slide, then you're ready to add a cover slip. Typically, cover slips can either be glass or they can be a film type. If you're using an automated cover slipper, typically if you're doing it by hand, you're going to be using a piece of glass. And so you want to put your media on top of your slide and then go ahead and just put the glass cover slip on top. Typically, you want to kind of lean it down. So you want to put it at an angle and just kind of lean it onto the slide. Because if you kind of just drop it on there, you're going to get air bubbles. And air bubbles are not great because the refractive index is so different than the tissue or the media or the cover slip. And so that makes it hard for the pathologist to see things. And also trapped air can act as an oxidizing agent. So that can kind of wear away your stains over time. So if you're going to cover slip manually, or if you're checking the cover slips from an automated cover slipper, you're going to want to look for air bubbles. And that's about it for this chapter. It was kind of a big chapter, but we also got to do some drawing, so that's cool. Maybe you watched me draw, which is also fine. After this, we're going to get into special stains. So specifically in Chapter 7, that's going to be carbohydrates and amyloid. So I look forward to going over that with you. And I hope you had a good time. As always, feel free to contact me with any kind of feedback. If you feel like maybe I missed something or maybe I misspoke, absolutely get a hold of me. If you feel like this is an engaging way to go about this, please let me know that too. So feedback, good or bad, get back to me with that. And as always, have a great one.